just finished talking about how microtubules get polymerized. So the alpha-beta tubulins coming together when activated with GTP in the beta tubulin come together and they form the protofilament, which then you have more protofilaments coming together, forming a sheet, rolling up, and then eventually becoming the microtubule. And then we talked about on the microtubule how we have the plus and minus end. Now what we're going to be seeing is that certain microtubules are going to be anchored at the minus end to what we call an MTOC, a microtubule organizing center. So the minus end of the microtubule will be anchored, and we will actually see assembly and disassembly happening all at the plus end. So again, GTP bound to the beta tubulin coming in and being added to the plus end of the microtubule, and then eventually that GTP in the beta tubulin gets hydrolyzed to GDP, and then that's subject to coming off. Now what we're going to look at is that there are three proteins that are called destabilizing proteins that help disassemble or depolymerize the microtubule at the plus end. So again, what these destabilizing proteins have in common is that they're all looking for GDP beta tubulin, and they're all activated, the proteins themselves, by ATP. Okay? So the three proteins, the destabilizing proteins we're going to talk about just now in brief, is kinesin 13. Kinesin 13 is similar to the kinesin motor protein, except it contributes to the depolymerization or disassembly of a microtubule. The other two is stathmin and catenin. Okay? So what we're going to see now in brief, and what's pictured here, is exactly how kinesin 13 and stathmin actually take apart the microtubule. So kinesin 13, I know this is a little difficult to see, but kinesin 13 is this tiny motor protein which has a tail and then has two heads. So the head region, just like with myosin, has an ATP binding site and then a site that binds to the microtubule. It so happens that with kinesin 13, the two heads on the kinesin 13 bind to both the alpha and the beta tubulin dimer. So one binds to the alpha, one binds to the beta. And again, the betas have GDP on their nucleotide binding site. So now they're subject to coming off. And kinesin 13 will basically bind to an alpha-beta dimer, and as long as the beta part of the dimer has GDP, breaks off the dimer. Now, with stathmin, it's a little bit different. Again, stathmin is activated by ATP, and it too is going to disassemble the protein or the microtubule at the plus end. But stathmin is a little bit of a longer protein and has the ability of breaking off a pair of these alpha-beta tubulin dimers. So here again we have the stathmin binding to the microtubule that's about to disassemble. When microtubules are about to disassemble, they splay. Okay, so they kind of open up like this. Okay, and so each of the individual protofilaments that make up the microtubule basically are all bending in preparation to break apart. And then stathmin is going to come in, and like I said, it binds to 4 of the alpha beta, or 2 pairs of alpha beta tubulin dimers, and then simply snaps them off. Okay, and then catenin works the same way. Now the next thing that I want to talk about then too, which I'm including in this particular shot, is drugs. So how certain drugs can be used to our advantage and how they affect the microtubules. We're going to look at 3 drugs specifically. 2 of these drugs contribute to depolymerizing or breaking down microtubules. So again, with the cytoskeleton proteins, if you have too few microtubules or not enough microtubules, the cell will die. Okay? Because again of the various roles that the microtubule plays. It helps move vesicles, it maintains the shape of the cell, and we're going to see later on that the microtubule plays a major role in mitosis. It's responsible for pulling the chromosomes apart during the anaphase stage of mitosis, which we'll be looking at in detail coming up in the next few episodes. So, two of these drugs that we're going to be looking at contribute to the depolymerization or breakdown of microtubules within the cell. Ultimately, these drugs are targeting the mitosis part, because if the cell cannot go through mitosis, then the cell will die. Two of them that we're going to look at just now, colchicine and nocadozole, or nocadozole I think is the proper pronunciation. Both of these are the ones that will contribute to the breakdown or disassembly of the microtubule. Now, these microtubules that the drugs are targeting, again, are involved with mitosis. These microtubules have what's known as dynamic instability. So what this means is that the minus end of the microtubule, which would be in this picture right here, is going to be anchored to the MTOC, or microtubule organizing center. A microtubule organizing center is simply a part of the cell where microtubules are growing from. So in the case of mitosis, the centriole is where the microtubules are going to be growing from in order to reach out and grab onto the chromosomes. Okay? So that means all the polymerization and depolymerization is going to be happening at the plus end. And it's known as a dynamically unstable or dynamic instability microtubule because the minus end is anchored, but the plus end of the microtubule is constantly growing and shrinking, until at one point it finally grows enough to actually bind onto the chromosome. Again, we'll be looking at this mechanism in a little bit more detail in a couple of episodes, but I'm just trying to give you a little heads up of what's to come, and then in order for you to understand how these drugs are working. So now that you understand that a little bit better, hopefully, so again, all the action is happening at the plus end. Because remember, when you go through the assembly, you're going to have GTP beta tubulin binding at the plus end, but then eventually all of that GTP beta tubulin gets hydrolyzed to GDP beta tubulin, and then that's going to be subject to the disassembly, right? And what's known as catastrophe, which leads to disassembly. Right? So now, colchicine and nocadozole. How do they work? What are they doing in order to contribute to this breakdown? Because remember, these two drugs, again, contribute to the depolymerization of microtubules. So colchicine, which is again found in the vinca plants, blocks the plus end of the microtubule. So basically, once a microtubule is going through the assembly process, so GTP beta tubulins hooking up to the plus end of a microtubule, what this drug then does, the colchicine, when it enters the cell, it will basically cap the plus end of the microtubule. So by capping off the plus end of the microtubule, you're going to do two things. One, you're going to prevent any more GTP beta tubulins from being able to bind. And the second thing is that all of the GTP beta tubulins that managed to get onto the microtubule's plus end are going to eventually hydrolyze the GTP to GDP and then be subject to falling apart. And that leads to the depolymerization of the microtubule. Nocadozole does the same exact thing. It's going to block the plus end of the microtubule, therefore causing all GTP beta tubulins at the plus end to go into the GDP form and then fall apart. And again, nocadozole is used to fight athlete's foot, right? Athlete's foot is basically a fungus that builds up on your feet when you haven't changed your shoes or your socks for a long time, right? And it's going to, again, block mitosis. Because if you don't have enough of these microtubules going through the assembly process, then those microtubules are never going to reach the chromosomes in order to pull them apart during anaphase of mitosis. So therefore, if the cell can't go through mitosis, it can't replicate and it can't duplicate. Okay? So that's how these two drugs work, colchicine and nocadozole. The third drug is taxol. So taxol, as I mentioned before, does the opposite of colchicine and nocadozole. Taxol is going to cause overpolymerization, so overgrowth, basically, of microtubules. Now, how does that work? So taxol is not going to target the microtubule filament itself, but it's going to target the building blocks of the microtubule, the alpha-beta tubulin dimer. So here's our picture here that we saw earlier of the alpha-beta tubulin. Remember, there's a GTP always in the alpha that's simply serving a structural role. And then in the beta, we have the GTP slash GDP illustrated here, because remember, the GTP eventually hydrolyzes to GDP. And then there's a position here, so here in green, where taxol can fit. Now notice that taxol is designed in such a way that it's going to bind into the beta subunit of the alpha-beta tubulin dimer. Now when taxol binds to the beta subunit, it's basically going to prevent hydrolysis from occurring. So it's going to keep GTP on and activated in the beta subunit and prevent it from being hydrolyzed to GDP. So therefore, if you have GTP always locked on, not going through hydrolysis in the beta subunit of the alpha-beta tubulin dimer, then you're going to have constant growth or constant assembly of microtubules. So your microtubules are going to continue to grow and grow and grow. And eventually, they're going to grow so much that it's basically going to overcrowd the cell and freeze up the cell. And again, this is happening during mitosis. So again, right around maybe about metaphase to the anaphase point, when the microtubules are growing and trying to capture the chromosomes in order to pull them apart, well, they just keep growing. They might bind to the chromosome, but they're still constantly growing. They're never shrinking, because it's the shrinking or the depolymerization of the microtubules that contributes to being able to pull the chromosomes apart. But if the microtubules are always growing, then you're going to overcrowd the cell and it'll freeze before it can go through or even be able to go through anaphase. And this is obviously going to kill the cell. And taxol is used to kill cancer cells, because cancer cells are basically cells that are proliferating or replicating out of control. So they're constantly going through mitosis. So taxol is going to target the beta tubulin of the alpha-beta tubulin dimer, help to lock on the GTP in the beta tubulin, and therefore keep the microtubules constantly growing and prevent them from ever disassembling or depolymerizing. Okay? So that's our take right now on microtubules. So there's a video previous to this on how microtubules get made and assembled. So we talk about the alpha-beta tubulin dimer, how they come together to form a microtubule, and then now we're going to start going into some of the roles then of the microtubule.